Hello, everyone, and welcome to our module on autonomic receptors. Before we talk about autonomic receptors, let's talk briefly about the neurotransmitters used by the autonomic nervous system. The two major autonomic neurotransmitters are acetylcholine and norepinephrine. Both of these neurotransmitters can be found in nerve endings of the autonomic nervous system, and when they're found there, they're stored inside these little sacs called vesicles. This is a picture of what they look like on the slide here. And so what happens when the autonomic nervous system is activated is nerves will depolarize, and this will cause calcium to come inside of the nerve ending. That's what's shown right here, calcium influx into the nerve. Calcium then is going to trigger these vesicles to release their neurotransmitters into the synapse. And that's what's shown here, the synaptic cleft. Neurotransmitters are being released by vesicles into the synaptic cleft. And this process requires calcium influx, which is triggered by the depolarization of neurons. Let's talk in a little bit more detail about the two autonomic neurotransmitters. We'll start with acetylcholine. So acetylcholine is synthesized from molecules of choline and acetyl-CoA. The enzyme that carries out this reaction is called choline acetyltransferase, and it synthesizes molecules of acetylcholine. Now, after acetylcholine has been released into the synaptic cleft, it's broken down by an enzyme called acetylcholinesterase. This enzyme breaks down acetylcholine into molecules of choline and acetic acid. And this is important in organophosphate poisoning, which I talk about in other videos. Organophosphates are poisons that inhibit acetylcholinesterase. That leads to very high levels of acetylcholine and overactivity of the parasympathetic nervous system. There's a tie-in here with infectious disease, and it has to do with botulinum toxin. So botulinum toxin is a toxin produced by certain bacteria, and what it does is it inhibits acetylcholine release from nerve endings. The major effects have to do with skeletal muscles, so nerve endings release acetylcholine to tell muscle fibers to contract. So when a person has botulism and botulinum toxin present in their system, they can't release acetylcholine onto nerve fibers, and as a result, they can't contract their muscles. And this leads to flaccid paralysis. And I talk in much more detail about botulinum toxin and botulism in videos in the infectious disease section. Okay, the other major neurotransmitter of the autonomic nervous system is norepinephrine, and it is synthesized from tyrosine, which is an amino acid. This is its structure on the slide here. Tyrosine is converted into a molecule called DOPA, which stands for dihydroxyphenylalanine, and that in turn is converted to dopamine, and then dopamine is converted into norepinephrine. So key things to remember here, especially for your board exams, are that norepinephrine comes from the amino acid tyrosine. The release of norepinephrine from nerve endings is modified by many drugs. I'll mention some of them here. Many of these I talk about in other videos. The first drug is tetrabenazine. What tetrabenazine does is it inhibits the vesicular monoamine transporter. This is a transporter that pulls dopamine in nerve endings inside of vesicles. So when you inhibit this with tetrabenazine, dopamine doesn't get stored inside of vesicles and it gets broken down in the cytoplasm. So what this does is it decreases the release of dopamine from neurons and also norepinephrine. This is really a niche drug. Its main use is in Huntington's disease, which is a neurologic disorder associated with excessive dopamine activity in the central nervous system. Cocaine is another drug that affects norepinephrine release from neurons. It blocks the reuptake of norepinephrine. That's what's shown here in my diagram. And this causes lots of norepinephrine activity throughout the body in the central and peripheral nervous system. And I talk about cocaine in the videos on adrenergic drugs. Tricyclic antidepressants are older antidepressants, but what they do is they block the reuptake of norepinephrine, and that increases norepinephrine activity. That's what's shown here. And then amphetamines, which I'll talk about in a minute, boost norepinephrine activity through a couple of different mechanisms, and I'll talk about this on the next slide. You should also be aware that norepinephrine can be administered intravenously as a drug. This is done in patients who have shock. The reason this is done is because norepinephrine is an alpha-1 agonist, so it's going to bind to alpha-1 receptors and contract smooth muscle in arterioles. That's going to raise the peripheral resistance and raise the blood pressure, and that's why it's a useful drug in shock. I'll briefly talk about amphetamines. So amphetamines are stimulants. They have clinical uses, but they're also drugs of abuse. And what they do is they increase the activity of dopamine and norepinephrine in the central nervous system, and that's why they are stimulating or stimulant drugs. They achieve these effects through a couple of mechanisms. First of all, they compete for the reuptake of dopamine and norepinephrine at transporters in nerve endings. So if you look at my picture here, this is norepinephrine being released, and then it's picked back up from the synaptic cleft and brought back into nerve endings by transporters. And so what amphetamine does is it competes for these transporters, and so norepinephrine stays in the synaptic cleft. It also displaces dopamine and norepinephrine from vesicles inside of nerve endings so that it spills into the cytoplasm. And when it gets into the cytoplasm, it's transported into the synapse. And so that's what's shown here. Amphetamine promotes the release of norepinephrine in addition to blocking its reuptake. And in the psychiatry section, I have videos on drugs of abuse, and I talk about amphetamines in much more detail there. Okay, so now that we've talked about neurotransmitters, let's talk about autonomic receptors. So the major receptors used by the autonomic nervous system are, first of all, nicotinic receptors. These receptors bind the neurotransmitter acetylcholine. Also muscarinic receptors. These receptors also bind acetylcholine, and there are many subtypes like M1 all the way through M5. And then finally, there are adrenergic receptors. These receptors bind norepinephrine and also epinephrine, and they have subtypes like alpha-1 and alpha-2 and beta-1 and beta-2 and beta-3. Now, in other videos, I talk about the physiologic and clinical effects of activation of all these different receptor types here. What we're going to focus on in this video is what the receptors actually cause to happen on a cellular level when they bind their neurotransmitter. So the easiest autonomic receptors to understand are the nicotinic receptors, so let's talk about those first. So nicotinic receptors are ligand-gated ion channels. So what does this mean? So a ligand is something that binds to a receptor. In this case, acetylcholine is the ligand that binds to nicotinic receptors. And so when acetylcholine, the ligand for these receptors, binds, ion channels open up and allow ions to move in and out of cells. And this is what's depicted in this slide here. The ligand, meaning acetylcholine, binds to its receptor here. And when that happens, ions begin to move in and out of cells. The ion channels open after the receptor binds acetylcholine, just like what's depicted here. And this causes an influx of sodium and calcium into target sites, into the tissue beds where the nicotinic receptors are found. So basically, acetylcholine binds the nicotinic receptors, the ion channels open, ions enter into the target site, and this depolarizes the target site. This could be another neuron if we're talking about the autonomic nervous system. It could be a muscle fiber if we're talking about the somatic nervous system and skeletal muscle. But whatever the target is, a neuron or a muscle, what causes the target to generate an effect is the depolarization of that target, and that's caused by these ion channels opening up when the nicotinic receptors bind acetylcholine. In contrast, the muscarinic and adrenergic receptors are more complicated to understand. So these receptors are called G-protein coupled receptors. So what does this mean? Well, they're called G-protein coupled because they're protein receptors attached to guanine nucleotides, like GDP and GDP. That's where they're called G-protein coupled. And when they bind to a neurotransmitter, they activate a second messenger system, and that's what carries out the effects on the target cell. So unlike nicotinic receptors, they don't simply open an ion channel and cause the target cell to depolarize. Instead, what they do is they trigger a cascade that leads to some second messenger generating the desired effect on the target cell. The two major second messengers of the autonomic nervous system are cyclic AMP and inositol triphosphate, also called IP3. And so the idea here is that when the autonomic nervous system wants to generate an effect on a target cell, it will release a neurotransmitter that will bind to one of those G-coupled protein receptors, and in turn, those receptors will boost the levels of either cyclic AMP or IP3 inside of target cells, and those are the second messengers that will generate the desired effect. So for cyclic AMP, what happens is when one of those G-protein coupled receptors is activated, that in turn activates the enzyme adenyl cyclase. It converts adenosine triphosphate, which is ATP that you're probably familiar with, into cyclic adenosine monophosphate, and this becomes the second messenger that goes on to generate the desired effects. The way IP3 is generated is depicted here in this image. So when the G-protein coupled receptor is activated from binding a neurotransmitter, it activates an enzyme called phospholipase C. Phospholipase C breaks down a structure called PIP2, which is phosphatidylinositol 4,5-bisphosphate. You don't need to know this long name, but it's a component of the cell membrane. And so phospholipase C takes PIP2 and breaks it down into IP3 and a second structure called DAG, which is diacylglycerol. And then IP3 is the second messenger. It generally increases calcium levels inside of cells. DAG can also be a second messenger. What it does is phosphorylate target proteins. But the idea here is that activation of this G-protein coupled receptor by a neurotransmitter leads to all these downstream effects, and then IP3 and DAG become second messengers that carry out those effects. So now that we understand the second messengers, let's go back and talk in more detail about the different G protein coupled receptors used by the autonomic nervous system. There are three subtypes of these G protein coupled receptors that you find throughout the body. They're called the GS, the GI, and the GQ subtype. Each of them has a different second messenger that generates different effects and actions on target cells. The GS subtype is what you find associated with beta 1 and beta 2 receptors. The GI subtype is associated with M2 receptors, and the GQ subtype is associated with alpha 1 and M3 receptors. This is a lot of confusing stuff to remember. Try to just get the big picture now, and then when you're closer to your exam, that's when you want to memorize exactly which G proteins go with which receptor types. So the GS subtype of G protein is called G sub S because it stimulates adenyl cyclase, and by doing this, it raises the intracellular levels of cyclic AMP. This is depicted here in this image. So the neurotransmitter, the ligand, binds to the G protein coupled receptor shown here. That in turn boosts the levels of cyclic AMP, which is the second messenger that goes on to generate the desired effects of the neurotransmitter binding the GS subtype G protein coupled receptor. So what does cyclic AMP do when its levels rise from a neurotransmitter binding a GS G protein coupled receptor? Well, in cardiac myocytes, those cells are stimulated by high levels of cyclic AMP inside the cell. And this is how beta 1 receptors work. I just told you they are linked to the GS subtype of G protein receptor. So for example, dobutamine, which is a drug that's a beta agonist, raises the heart rate. The way it raises the heart rate is by binding to a beta 1 receptor in the heart, activating that receptor and boosting the levels of cyclic AMP inside the cell. In vascular smooth muscle, like you find around arterioles in the peripheral circulation, cyclic AMP levels, when they go up, they relax smooth muscle. This is how beta 2 receptors work. They are also linked to the GS subtype of G protein coupled receptor. So once again, let's consider dobutamine, the drug that's a beta agonist. It's going to bind to beta 2 receptors and relax vascular smooth muscle by raising the level of cyclic AMP inside of smooth muscle fibers, and this is going to lower peripheral resistance and lower the blood pressure. So an easy way to keep this straight is to just remember that all beta receptors are linked to the GS subtype of G protein coupled receptors, and therefore all beta receptors, when they bind to norepinephrine, are going to increase intracellular levels of cyclic AMP. And these details right here are the kind of things you're going to be asked about on your board exams. Next, let's talk about the GI subtype of G protein coupled receptors, and they're called G sub I because these receptors inhibit the enzyme adenylyl cyclase, the opposite of the GS receptors. So when these receptors bind a neurotransmitter, that's going to lead to decreased intracellular levels of cyclic AMP. And the main place where this is important is in cardiac myocytes. So binding of a neurotransmitter to a GI receptor inhibits cardiac myocytes, which decreases contractility. And this is the way M2 receptors work. So M2 receptors are part of the parasympathetic nervous system, and when they bind acetylcholine, that leads to activation of a GI subtype of G protein receptor, and that's going to decrease the levels of cyclic AMP and inhibit cardiac myocytes and reduce contractility. You also find the G sub I subtype of G protein receptor in alpha 2 receptors, which I talk about in detail in other videos. So here's a summary slide of how the GS and GI systems work. This is a confusing slide here, but I'll walk you through it. And if you understand this, you can answer lots of questions about how these beta 1 and beta 2 and M2 receptors work. So let's start by considering what happens when a beta 1 receptor gets activated by norepinephrine. Beta 1 receptors are linked to GS proteins, which activate adenyl cyclase. That's going to boost the levels of cyclic AMP. Cyclic AMP is a second messenger. It's going to activate an enzyme called protein kinase A. Protein kinase A is going to trigger more calcium to come inside of cardiac myocytes. And anytime you bring more calcium into cardiac myocytes, more calcium gets released from the sarcoplasmic reticulum, and therefore you get more vigorous contraction. And so this is how binding of norepinephrine to a beta 1 receptor increases contractility in cardiac myocytes. The reverse happens when acetylcholine, as part of the parasympathetic nervous system, binds to M2 receptors on cardiac myocytes. Those are associated with G sub I proteins, and that's going to do the opposite of what the GS protein did. And then in vascular smooth muscle, depicted over here, we talked about how binding of norepinephrine to beta 2 receptors relaxes vascular smooth muscle. And this is done through a GS protein, which activates adenyl cyclase and boosts the level of cyclic AMP. But what the second messenger cyclic AMP does in vascular smooth muscle is different from what it does in cardiac muscle. In vascular smooth muscle, cyclic AMP inhibits myosin light chain kinase. And you need myosin light chain kinase to interact with calcium and calmodulin in order to cause contraction of smooth muscle. So when you inhibit this, you're going to get less contraction and relaxation. And that's how beta 2 receptors generate relaxation of vascular smooth muscle. Okay, our last subtype of G proteins are the GQ subtype. These receptors activate phospholipase C and increase IP3 levels, that other second messenger we talked about earlier, different from cyclic AMP. The major place where you find GQ receptors are associated with vascular smooth muscle, and when they bind a neurotransmitter, these receptors generate contraction of vascular smooth muscle. So for example, alpha-1 receptors are linked to the GQ subtype of G protein receptor, and when they bind norepinephrine, they lead to contraction of vascular smooth muscle. This is how norepinephrine raises your blood pressure by contracting smooth muscle around arterioles. In the parasympathetic nervous system, the GQ subtype of G protein receptor is how M3 receptors work. When these receptors bind acetylcholine, they work through a GQ system to generate vasoconstriction, bronchoconstriction, contraction of the detrusor muscle in the bladder, or contraction of the ciliary muscle in the eye. All of these processes require contraction of smooth muscle, and that's done through M3 receptors linked to a GQ subtype of G protein coupled receptors. So the GQ subtype of G protein coupled receptors is only found in vascular smooth muscle, and when it binds a neurotransmitter, this leads to contraction. The way this happens is depicted here in this slide. So if the GQ protein binds a neurotransmitter, it activates an enzyme called phospholipase C, which we talked about earlier, which takes PIP2 and converts it to IP3. This is the second messenger of the GQ system. IP3 will trigger calcium release from the sarcoplasmic reticulum inside vascular smooth muscle cells, and that leads to contraction. So shown on this slide is a table with lots of different receptors found throughout the body and which G protein class they are associated with, either Q, I, or S. This is really annoying stuff to have to remember. I don't recommend you memorize any of this until right before your exam because you'll quickly forget it. Just make sure you understand how these systems work in general, and then later you can commit to memory that the alpha-1 receptor is associated with GQ or that the dopamine 1 receptor is associated with G sub S. These systems are also associated with histamine receptors, vasopressin receptors. All different kinds of receptors throughout the body have these different GQ, GI, and GS systems. And here's another table showing the 3 different G protein subclasses and all the receptors that they are linked to. There are lots of mnemonics you can use to remember these. You can use whichever one you want. You can find lots of these online. One that many students use is Have1M&M. This stands for all the receptors linked to GQ. MAD2 is for all the receptors linked to GI, and then all the others are linked to G sub S. And then finally, here's some take-home points about how the different nervous systems of the body work. So the sympathetic nervous system uses norepinephrine as a neurotransmitter that binds to alpha and beta receptors. And then those different receptors are linked to different G protein subtypes. Parasympathetic system works through acetylcholine and muscarinic receptors, and those receptors are also linked to different G protein subtypes. And then finally, the somatic nervous system, which is how we contract voluntary muscles like skeletal muscles, that works through acetylcholine on nicotinic receptors, and those receptors are much simpler to understand. They are ligand-gated ion channels like we talked about earlier. So what they're going to describe on your exams is either a drug or a pathologic process that activates the sympathetic nervous system or maybe the parasympathetic system, and then they're going to want you to know which of these receptor systems is activated in those processes or associated with those drugs. And so that's the kind of stuff I put on the last 2 slides. It's very detailed and hard to remember. Best thing to do is just get the big picture right now, and then when you're close to your exam, memorize all those different receptor types and which G protein they're linked to. And that concludes our module on autonomic receptors.